Welcome, everybody, to The Animal Heartbeat. We are very excited to bring you this episode. Jose Novomatos and I, Kieran Bourget, are your co-hosts. And our guest today, we are honored to have him with us. He's Professor of Cardiology and Associate Dean of Research at North Carolina State University, Joshua Stern. Thanks, Josh, for joining us so much.
Thanks for having me, folks.
Jose and I are both obviously interested in feline cardiology. We have a joke on The Animal Heartbeat that whenever Jose talks about feline cardiology, it's like a drinking game. So, people are waiting for that to happen. Jose, why don't you indulge the listeners and begin talking about feline heart disease?
Yeah, this is already my favorite podcast ever. Welcome, Josh, and thanks very much for joining us. So a lot of exciting stuff to talk about, but we thought we would start with the basics. So hypertrophic cardiomyopathy is defined as left ventricular hypertrophy. So LVH is the hallmark of HCM. Can you tell us a bit about what causes LVH? Why would a sarcomeric disease, why would a sarcomeric mutation end up in a thick ventricle?
I think we're learning a little bit about this all the time. And as we learn more about cats in particular, I think we'll probably uncover some specific things that have to do with how mutations in cats actually lead to the wall thickening. But what we know about this disease in people is that many different sarcomeric mutations kind of all end up in the same disease phenotype of that characteristic wall thickening. And most of those mutations are in the sarcomere, the most basic contractile unit of heart muscle. And what we see kind of functionally or ultra functionally when we get right down on the sarcomere is that these sarcomeres in cats and in people and in mouse models are hypercontractile. So they're doing their job at an exceptional rate. So there's more kind of heads of the myosin sticking up and engaging and squeezing down or cycling. And that hyperactive sarcomere is kind of the target of a lot of novel drug therapies. You can kind of think of it as, if the sarcomere is working too hard, it's squeezing, it's going to the gym a little bit more and more every time, and it's getting thicker. And so that's, I think, the functional basis, or at least the most broadly accepted functional basis of wall thickening in HCM.
Well, what is interesting is one would think, okay, so that's the main target for us to treat HCM. So we should target LVH. But interestingly, that has not been the case in the past maybe 20, 30 years. We have not necessarily have been focusing on LVH, which is changing, and we'll discuss that in a second. So besides left ventricular hypertrophy as a target for these diseases, which other targets do you think could be amenable or interesting to address with HCM? And maybe more importantly, at which stage we would ideally address those changes?
I mean, I think for decades, we've really focused on outcomes of HCM and what we can do to either slow down or prevent those outcomes, like most cardiac disease, particularly in veterinary medicine. And this is, I think, a change in approach for a lot of different diseases across humans and animals is we're really moving more and more towards targeted therapies that work to understand the disease biology and pathobiology of what's actually causing the ultimate disease itself. I think, from a feline cardiology perspective, I personally want to ensure we have something on the market that stops all cats from developing blood clots, because I think that's a devastating outcome. If we had something that we were certain did that well, and we never had to worry about the disease outcome, that would be a great target. Although not as common as heart failure or maybe not sudden cardiac death.
But just so awful, isn't it? I mean, as an outcome, we all experience that, that sort of emotional side of it, and just see the animals when, they're affected by an AT. It's just such a shocker that it'd be amazing to have something that we knew was super effective there.
Yeah, I think so too. I think the challenge for me when I think about these things is often the bit of information that's missing is maybe not so much the pharmaceuticals as is the prediction part. And so for me, we have some pretty decent drugs that work to prevent thrombosis. We just need to know when to start them and when to expand them. But if there was, a silver bullet there that just, you didn't have to worry about it, you just started all cats with HCM on it, it was completely safe and completely effective, that would be a huge win for cats and everybody else. So that would be a good target. I think, we could probably do better on congestive heart failure in many ways. I think the jury for me is still out on whether there's some benefit to effectively blunting the renin-angiotensin-aldosterone system. I don't think we have much evidence that we do that effectively, and so
I think the data we have on that in cats is weak, isn't it? It's difficult to draw strong conclusions from it. Yeah,
certainly. I think with the propensity for kidney disease in cats, if we could figure out a more suitable diuretic that didn't exacerbate that, we might be in better shape. So I think there's probably a lot of good stuff coming down the pike in all of those areas for cats that could be crossed over from more recent human trials and things like that.
Something that I think is always very interesting is, so obstructive HCM, SAM, is a very big thing in humans. People with obstructive hypertrophic cardiomyopathy have severe clinical signs associated with increased morbidity, mortality, and is the target in human HCM. Feline HCM mirrors so well the human disease in every aspect, from clinical signs, histological findings, echo, everything. Is it different? Is obstructive HCM different in cats versus humans, or we just don't know?
That is something that keeps me up at night. I think it's a really
we often worry about chest pain and HCM.
Yeah, exactly. That's a really great question. The thing about obstruction in cats, to me, is that I think we mislabel that so frequently that when we talk collectively about obstructive HCM, I think we're including a lot of cats that are not truly obstructed. And so I think the sense we get about how cats with obstruction behave is somewhat different than if we were talking about humans with obstruction. I guess, to me, a lot of the cats that get an echo pop up as having moderate obstruction at home on their couch are likely not at all obstructed. And I would venture a guess that that's probably, 75 or 80% of the cats I see that get the label of HOCM on their echo. When I'm reviewing studies, I don't think that's a high enough gradient for that cat to really be persistently obstructed. When you look up and the echo report shows the heart rate at the time was 220 and it was dynamic obstruction, and all of a sudden this cat needs a beta blocker.
So what's your cutoff for calling something obstruction? When do you reach for a beta blocker?
That's a great question. I don't know the answer to that question, actually. It is a sliding scale for me these days. I think I look at the cat overall and think more about it than I do based on a number alone.
Right.
To me, beta blockers are so benign in early phase of disease. I don't find much harm in their use. And so, do I reach for them in cats that are probably not persistently obstructed? I think I do. But when I'm trying to make the pitch to my human colleagues about drug trials that cats are just like humans, I think we probably need to be looking at those severely obstructed cats only and saying, this is what an obstructed cat looks like. I think it probably mimics an obstructed human better. And I think there's some evidence of that anecdotally in clinical cases, right? Like, you've probably both had the cat that was severely obstructed that you thought, does that cat have chest pain when it exercises? Is it limiting its exercise based on that?
And where you treat them with a beta blocker and they change.
Yes, exactly.
It's and that's sort of proofs in the pudding, isn't it? Or proof of the pudding is in the eating. And I think it's hard to, I don't know, it's hard to do that. I'm not sure there's a great protocol and certainly no consensus.
Exactly. And we're left with using those survey tools, which are just not the level of evidence one needs. If our cats could tell us that they feel a lot better, it'd be great. But for me, I think you could probably start a beta blocker, and if the cat doesn't seem to have an improved quality of life to the owner, probably wasn't severely obstructed to begin with. But I don't really know the answer to that question. And I think because of the kind of relatively benign nature of beta blockers at the doses we use in cats, people have just felt fairly comfortable saying, well, you're obstructed, reach for a beta blocker. And it's left a lot of people to have this impression that they don't do much, which is probably incorrect.
Interesting. So I think the closest thing that we have to a well-characterized controlled population of cats with cardiomyopathy to study is the population of cats that formerly was at UC Davis, that many people will be familiar with from Mark Kittleson's work and latterly your work, Josh. Now, I understand that you've brought them with you from UC Davis.
That's nice.
We moved 50 cats across the continent. Yeah.
Wow. God.
I cannot
How
imagine
much
the
gabapentin did
paperwork.
that take?
Yeah.
What have you learned from those cats? What advantages has that colony had for our understanding of HCM and natural history and treatment compared to the population of cats we're all seeing out in the wild?
Yeah, the cat colony has been a really incredible thing for us to study in terms of learning about HCM and being able to look at novel interventions that wouldn't quite be ready for prime time for a study in client-owned cats. I think, we have these cats that are genetically predisposed to having HCM, and we've learned a lot about penetrance of the genetic mutation. It mimics the human population so well, in a way. We have virtually no good prediction of when a cat will develop HCM, even if they are genetically predisposed and have 2 copies of the mutation.
Really?
Within the same family, 2 siblings. Yeah, 2 siblings can be a year apart in their onset of hypertrophy, which I think is very fascinating. If you take, absolute full siblings, they both have the homozygous variant that causes HCM in that colony, and their onset of thickening will be a year apart sometimes. And so I think I've learned a lot about this idea of expressing disease and how we go about ensuring that we're comparing apples to apples. And so our studies are, I think we're very fortunate to be able to still do small scale pharmacologic interventions because the cats and their environment are so well controlled. But we're still subject to the variability of this disease, which across the general cat population is massive. Even within this extended family is still pretty great.
Wow. I mean, it's fascinating and stimulating, but so frustrating, isn't it?
Yes. Yeah, absolutely.
What is interesting about, so that's something I think is indeed fascinating, the genotype-phenotype correlation. And in your colony, so I like to think that environment plays a role, that environmental factors will play a role on the phenotype and so on. But in your population, the environment is controlled. And so, as you were saying, the 2 siblings will be subject to the same environment. Do you think then are epigenetic factors, right? I think that's the only thing that is potentially missing to affect the phenotype. Have you been looking into that, how epigenetic factors, other mutations may affect the phenotype?
We've not gone down the rabbit hole just yet of looking at, CPG islands and methylation and all of the kind of nitty-gritty of other genetic factors that might influence expression of disease. We have looked at disease modifiers in the colony to some extent. We've whole genome sequenced every cat in our colony. Wow. We're not quite caught up on that right now. The youngest generation still needs to be sequenced, but every time we create a generation of cats, we work to make sure that we have them fully sequenced. And so we have looked for some modifying genes, and there are some targets in there that look like, maybe that's playing a role. The problem is there's only so much you can do at a time. And so it depends, what the real best use of these cats is and the most benefit we're going to get. And so we've moved mostly towards, at least in recent years, looking at novel therapies with the cats rather than looking at breeding experiments and testing out new genotype-phenotype correlates. And that's predominantly because we think that's what's going to get the most bang for our buck to help cats and people. But it is a super amazing resource that, if I had unlimited time and unlimited money, we could do a lot of things to try and study disease expression and how genetics really modifies that. We just haven't quite gotten there yet.
And I think unlimited cats, because each cat only has a given tolerance for its welfare, doesn't it? I mean, they can't be subjected to 4 clinical trials simultaneously. I mean, obviously, it not only messes with the clinical trial, but as an individual cat, that's just not okay. And I know that, from my understanding of the work that you've done, their welfare is super high in this colony of cats. So, and I think that's something to be really proud of. It can be hard to achieve that balance, I think.
Yeah, we're really, really proud of the care that these cats get. I mean, for us, we know all of these cats. We know them well. We know their behavior and their attitude. We know who needs to get their echo in the morning versus the afternoon because they get progressively cranky as the day goes on,
Uh-oh, Fluffy missed breakfast.
That's right. So we work hard to ensure that, the cats are being used in a way that really does look after their welfare. And so most of our cats will not go through more than one trial in a year period in terms of a pharmaceutical intervention. And then we have our ongoing background monitoring that happens where most of these cats will receive a echo, full lab work and everything with sample storage every 6 months or so. And in between that, they're just kind of happy cats that are living in their little cat trees. And that is probably the best part about having the cats, is knowing that we feel really good about how they're cared for. We're just about to do a round of spays and neuters and adoptions out of the colony, which is really great. We have a super good network for that here at NC State. And so we're excited to do that. One of them is about to become our library cat that gets to hang out here in the hospital. So we're super, super happy and fortunate to be in a position to do that and treat the cats that way. But it does, in order to maintain that, it requires a lot of cats. So the colony is relatively large and not cheap to maintain for sure.
You were saying that you have focused more now on clinical trials, which I think your research is a great example of One Health and then translation research from cats to humans and vice versa. In the past, I have been depressed with the fact that we did not know the cause of HCM in cats. We know 4 mutations in cats causing the disease, and so it depressed me a bit about how can we go ahead and treat these cats if we don't even know what is causing them. But it seems like these new treatment avenues that you have been working on and so on, it might not matter, right? It might not matter which mutation is causing the disease or what is the underlying cause, because you are addressing, hopefully, the mechanism will be the same. Is that right? Even though, of course, your population is very well genotyped and characterized, is it right that maybe the cause doesn't matter on these new treatments?
Yeah, I think that's true. I think, I think a lot of the drugs that we've worked with or have come to market recently were designed really to target the sarcomere and target myosin binding protein C3, which is one of the most commonly mutated genes in people with HCM, and just so happens to be the gene that's mutated in our Maine Coon and Ragdoll cats. So although those drugs were developed with maybe a specific target that we were sure they would likely work on, it does seem that what they're doing overall is kind of broadly targeting the kind of pathobiology of this disease. The same goes for the rapamycin compound that I'm sure we'll talk about in a bit. Again, broadly targeting maladaptive hypertrophy, not just a single mutation. I am also occasionally depressed about the lack of understanding what genetics are kind of causing the general cat population to have hypertrophic cardiomyopathy. But I do think we're getting a whole lot closer in that. I mean, I saw recently a preprint that came from the RVC. Probably some of you were on that. I didn't see the author list, but I know it came out of that group, looking at massive sequencing in a lot of cats with HCM and identifying some common variants. Similarly, my graduate student just finished a whole genome sequencing study of more than 100 cats with HCM and matched RNA sequencing. And so I think we're going to come out with something at the end of those 2 studies. If you put those together, it's a bigger whole genome sequencing study than most human studies looking at HCM.
Right.
I think we'll have some information there. And I think it's going to be that HCM in the cat is heterogeneous and additive and filled with common variants that when you link them all together, you get this disease. And I think that's really cool. And probably does underscore exactly what you're saying, that you don't need to worry so much about the therapy being super targeted to any one single cause.
I think there's always these stories you hear about how some of the breeding community get very, very focused on a mutation. And I think, of course, this has happened with the A31P in the Maine Coon and the R820W in the Ragdoll. Certainly in the UK, the Ragdoll breeding community became very focused, and they really didn't want to breed from anything with a single copy of the gene, or obviously the homozygous ones, but they didn't breed from anything. And then, of course, they end up in a situation where they massively narrow the gene pool. There's potentially a history of some introduction of other diseases from Persians maybe being crossed in with those ragdolls.
Yep.
And it's so challenging. So I think part of the challenge with the genetic research is how we communicate that and get that out there to people, and make sure that we kind of moderate that message, if you like, and make sure it's the whole picture. This is how we can use it, and this is how we should use it. I mean, that doesn't guarantee everyone does what we ask them to do, but hopefully we can moderate things a little more.
Absolutely. And those studies can really get away from you, right? Once you put something out there, it's out there, and it will be used however the public is going to use it. And so, if you look back at the A31P mutation in myosin protein C, when that came out, the original publication in those cats out of Mark Kittleson's lab was that this is a straightforward autosomal dominant disease. Every heterozygote individual got the disease. The homozygotes got more severe disease. It was done and dusted and pretty clear cut. You look 12 years later in our colony, and it behaves more commonly as though it's recessive. And so, the heterozygotes may get disease, but they don't necessarily get disease at a higher rate than the average cat, house cat. And so you have to wonder whether or not, what's going on in those different studies, but it's too late because, the Maine Coon breeders and the general public have taken the information that was originally published, and they've worked to eliminate even heterozygous variants for that mutation. And that's where you start to really significantly narrow the breeding pool. And often they get picked up before anybody's ever validated or talked about a mutation being real and important. I mean, you can look at HCM genetic variants that have been reported, and there have been a few reported recently that were complete bunk.
And labs are offering them as clinical tests. Yet there's no data to support them.
Tests. That's right. I mean, to be perfectly honest, I'm only sure of the A31P and the R820W as true causative variants. I'm not sure of anything else at this point. I think all of the rest warrants a lot of continued work. And I think many of what we know in terms of genetics are population restricted as well. So should you apply a test that's identified in a Canadian cohort of dogs or cats to a UK cohort? Probably not.
And looking at the stuff from boxer dogs and dilated cardiomyopathy in Dobermans, that absolutely pans out, doesn't it? There are geographic differences there in whether those mutations are important or not.
Yeah, it's fascinating. And so clearly there's genetic architecture there that we don't understand. Are there protective loci? Are there modifiers that must be found in combination with these variants for them to be important? We really don't understand it. All we know is what we see in the clinic. And for me, I think that's where these tests start to really become important. So let's just go to the clinic and say, does this test inform me about something from a clinical perspective? Either risk of disease, severity of disease, need for intervention. That's when I start to care about them the most.
We have talked a lot already about myosin inhibitors, small molecule inhibitors in previous, not a lot, but in previous episodes, which is incredibly smart drug. Is so cool and so exciting that a tiny drug can act at the myosin head and so on. How outstanding is that? So you studied two of the main drugs in the market, mavacamten and aficamten. Can you tell us a bit, how did that happen? And what were the main results from your studies, and how did that help them to go ahead and open the doors for RCTs in humans?
That's a great question. We really have, I think, a long history of working with the biotech and pharmaceutical companies, largely from the Bay Area. So I was well positioned at Davis with the colony to meet some incredible scientists working in cardiovascular health at a few different companies. The first experience I had was with a company called Myocardia. Myocardia now is Bristol Myers Squibb, but at the time they were a relatively small collection of passionate scientists working to address a kind of pharmaceutical desert of HCM therapies. And they rounded up a really incredibly brilliant team to work on that. And one of the gaps in knowledge that they had was around a suitable model organism, because mice just don't recapitulate HCM in the ways that humans do. In particular, obstructive cardiomyopathy is not really seen in the same way in a lot of model organisms. And so, we were at the time publishing intermittently on hypertrophic cardiomyopathy in the cat colony, but probably hadn't really made it to their radar in terms of these cats existing. And then, as luck would have it, you talk enough about something to enough people that are interested, those kind of grassroots connections happen. And a colleague of mine connected our team with a scientist at Myocardia. And that really took off, because he said, gosh, this is probably the most suitable large animal model we've ever seen to mimic HCM. Let's see if this compound that we have does what we think it should do. And thus began a really fruitful relationship that became more and more trials with more and more companies as they started to see the value of the colony be demonstrated. That very first study that we did was with an early iteration of what would later become mavacamten. And that early iteration, we simply did a injectable acute dosing study showing that the drug alleviated obstruction and had kind of on-target effects at the level of the heart function, and that the dose response was predictable. And that was enough to endear the cat colony to the pharmaceutical industry like that.
Nice.
It's a great story of a lead in to that collaborative research, isn't it? Yeah.
Yeah. And now we work with many companies on many different compounds around this area. And it's been fantastic. We, in some ways, function as a symbiotic CRO. So we work a little bit like a contract research organization in that I'm happy to have a pharmaceutical company come to my door and say, we'd like to show that this compound does this in cats. But then the symbiotic part is I say, but this is what I want to know in order to help cats. And
right. So it's common goals, isn't it?
Yeah. So let's broaden the scope a little bit and make sure that we all enjoy this and get what we want out of it. And that has been the highlight so far of my career, is working with these compounds and these companies and getting my graduate students to experience what that side of science is like has been really phenomenal.
That's great.
That's right. And they are receptive to that. When they come to talk to you, they are receptive to, that's nice. That's nice.
It's honestly, they've been some of the most rewarding scientific collaborations. When we do this, most of the time, they come out and work with us for those days. We do some really fun stuff. I think you've probably all seen, we've published some pressure volume loops using one of the myosin inhibitors in cats. And those are just things that, you don't do on a daily basis. And so super exciting to learn about what pressure volume loops in cats with HCM look like.
There's nothing I love more in cardiac physiology than a pressure volume loop. My resident she is gutted, about this, because I keep saying, "Oh, we've got to talk more about this. We got to talk more. Let's next time we come,
Yes.
in book club, let's talk more about pressure volume loops." And you can see her just dying a little bit inside.
Yes.
But I just think it's amazing to
Yes.
see what is happening in the ventricle, in the cat, and we see it on echo all the time. But to see that in this physiology sort of concept, which any of the listeners who've read, Levick or Katz or Opie or any of those books will be familiar with what they're talking about. You see this and it can seem quite abstract. But to me, it really helps me understand the theory of what's going on at what time in the disease. Yes. And then to be able to see that in reality, fantastic stuff.
Yeah, that was really lovely. It is one of the things that bothers me the most is that we haven't published it yet. So we're working on that right now, is writing up the PV loop study, because it was an add-on to a study that wasn't necessary for the goals of the company, and just generated some beautiful data that we certainly are committed to publishing, and we'll get out there as part of a thesis here coming up soon. But it was really lovely, and it also demonstrated to us, that was one of the times where it really clicked for me, was seeing the pressure volume loop that was characteristic of hypertrophic cardiomyopathy in the cats, just looking precisely like what we know in humans. Seeing that pressure volume loop normalize with one of the interventional compounds that we had.
That's so powerful.
That's so cool. That's so cool.
It was so cool.
Yeah, because I guess if you would say that initially, people would be worried that it would decrease contractility beyond what is normal and would affect contractility and so on. The fact that the drug can normalize the sarcomere.
Yes.
That's beautiful. That's beautiful. Yep. That's crazy. Yeah.
And I haven't published any of the ones. I haven't published any of the loops or shown any of the loops in conferences of obstruction, but because they just get so ugly. But that was another kind of light bulb moment for me, was look what happens when these cats begin to obstruct to this pressure volume loop, just how disruptive obstruction really is to the normal mechanics of the LV. Super fascinating stuff.
I have a couple of questions. And one actually slightly takes us back to something we were talking about previously. And one is what's next? The what's next is about mavacamten, aficamten. Where are those drugs going? Are we going to see them on the shelf in the veterinary pharmacy in the next 1-2 years? What's the story of those drugs to come? The second question you might want to answer first. If we know we've got such strong evidence that overzealous systolic function is a driving force behind the pathophysiology, the natural history of HCM in cats, why have we got some evidence of systolic dysfunction relatively early in hypertrophic cardiomyopathy that's out there in the literature? I'm thinking specifically, actually, of Ilaria Spaller's work on speckle tracking echo, wherein cats who were genotype positive, phenotype negative, so non-hypertrophic cats, they had evidence of systolic dysfunction, reduced strain. And I've always thought, it's super interesting, because maybe the systolic function reduces quite early in disease, and this is part of progression. Have you got any ideas? I know you might not have any data, but have you got any ideas as to why we're seeing these conflicting data? And I guess we've got to go on what you're measuring in the heart. You're measuring that systolic function is overzealous, and these small molecule inhibitors will normalize the pressure volume loop. That's gold standard. Why have we misinterpreted that on echo, or have we misinterpreted that on echo?
I think that is a fascinating conundrum. I think the work that shows kind of pre-echo positive alteration is really fascinating to me, and that's been mimicked in a few different ways, right? So Ilaria's work was beautiful in what it showed early on for genotype positive animals. The same thing was true in some work that we did in the colony looking at hypercoagulability. So genotype positive, phenotype negative cats still had overzealous platelet activation.
That's amazing, isn't it? Because we always look at the left atrium. God, the left atrium's big. That's a problem.
Yeah, but these cats had it before they even had thickened left ventricles. And so, what's really happening there in this kind of silent early phase of disease is a fascinating question to me. It would be, if you knew what cats were going to go on to develop HCM, what could you do at that stage of disease to keep them from ever developing classical HCM? That would be fascinating. The short answer to your question is I don't know what's going on there, but I would say I'm cautious when I speak with folks about these drugs and what they're doing. I'm cautious about saying that the change that we're making at the individual sarcomere is not necessarily what's going to be reflected at the whole heart level by echo. And we have to look at both of those things to see what's really happening and where we're really headed with these therapies. Now, in the case of the myosin inhibiting compounds, they do go in the same direction, right? So we know what they're doing at the level of the sarcomere because of the really lovely, elaborate pharmacology work that's been done. And then we know what they're doing at the level of echo, and we know what they're doing at the level of PV loops. And so those things are all going in the same direction. But I don't know why early on in disease you would get these signals of functional deficits or changes in function that seem somewhat nonsensical, unless this is some sort of a reflection of kind of the disease transition to more hyperactive. I don't think we know early on what's going on in terms of sarcomeric function.
And what's next for the Campton drugs, the small molecule inhibitors? What are they something we're going to see on the pharmacy shelves anytime soon?
Well, I would love to think so. I mean, mavacamten is now available for use in humans. Aficamten is in kind of final wrapping up of trials, it looks like. And so those will be available. I assume if we don't get to a point where we find one that can go through a true veterinary clinical trial, that people are going to pick them up and start using them, whether we like it or not. And we might learn something about that in a less controlled environment. What we really need is somebody to get us an oral compound of a myosin inhibitor that we can do true long-term clinical studies with. I would love to do a long-term oral Campton compound trial in our cats, at least 6 months of dosing, and look at safety and efficacy in that environment. I think that would be really important and needs to be done. And I think that would be the most appropriate lead in to using these in client-owned cats. But at this point, we don't have a compound that's been released for us to use in that way. And we've not been contracted to do any work that looks quite like that. So I would say somebody is going to have to either develop, which is possible on the pharmaceutical side in veterinary industry, or purchase a Campton compound that isn't mavacamten or aficamten, because those ones are so tightly regulated right now that I don't think anybody's just going to hand us one of those to use orally. But if you follow the literature, the first one I published was, I think, 461, and then 581, and then 586. That refers to the kind of version of the compound.
Yeah. So
there are hundreds of these compounds that have been raised up by these companies. And so, we would take second in line without be complaining too much.
How long is the patent? Do you know? Because these guys, of course, invested millions, billions, I don't know, building all this research. I remember, as you were mentioning, the first science paper in mice.
Yes.
It felt like that paper was 3 PhD theses in one paper. The amount of data and so crazy. So I can only imagine how much investment this has been going through.
Yeah, my understanding is that the patents kind of are dependent on indications that they get and how many of those they get and at what time they file for those indications. And so if you follow the literature, like mavacamten came out based on those studies, but there are other ongoing clinical trials of mavacamten that will likely, if they work in other indications, will likely result in extension of patents. And so I assume we'll be in the same boat we were in with pimobendan, where it is on patent for a really long time.
Yeah. Now that you mentioned that indication, so we talk about dynamic left ventricular outflow tract obstruction before, and of course the first clinical trials for mavacamten and aficamten and your data target outflow tract obstruction, which makes a lot of sense for many different reasons. And now, interestingly, there are clinical trials, Red Hood and others, that are looking at non-obstructive HCM, which I thought is really cool because it can play in our favor. As you were saying, we don't know if cats are obstructive or not, or we don't have really great tools to know which cats are obstructive or not. So we may say in the future, maybe it doesn't matter. It will work in obstructive and non-obstructive. Do you have any data on non-obstructive HCM?
Yes, we do. And in fact, you'll see this year at ACVIM, we're presenting a sarcomere active compound that was in non-obstructed cats, or at least not obstructed at the time of the study. Most of our cats in our colony are not obstructed. And we try for these trials to choose cats that are obstructed, but you'll notice in our methods that we often induce obstruction in these cats by bringing their heart rates back up into a higher zone. And so, I think most of our work can honestly be interpreted as though it's in non-obstructed cats. None of the cats, except for one or two that I can think of in my colony right now, would I actually be convinced are persistently obstructed. Although that's some work I'd love to do to prove that. But what's coming up at ACVIM, I think you could see as a non-obstructed work. And that's with an oral compound similar to aficamten, but not the same.
So obviously a lot of the interest from the human pharmaceutical companies in the Camptons is related to outflow tract obstruction. It's a relief of symptoms. It's potentially improves prognosis. One of the things that we'd like to know for our population of cats that we see that are non-obstructive that we've been talking about, would they benefit from anti-hypertrophic drugs more? And this brings us on to the RapaCat trial. And thinking about rapamycin. Tell us a bit about your work on rapamycin, Josh, and what your understanding of where we are with that drug, and should we be reaching for it now, or is more work needed?
Yeah, that's exactly where we're headed right now in our work, is really investigating what rapamycin is doing and how useful it is in the situation of occult cardiomyopathy. And so, to give you a little bit of a lay of the land, we've worked with rapamycin, a delayed release formulation of rapamycin, pretty extensively now, completing one clinical trial in client-owned cats and some work in the cat colony looking at kind of multi-omics of what rapamycin is doing. To me, rapamycin is really targeting the maladaptive hypertrophy that goes along with HCM. And if you follow on down the line from a physiology perspective, a lot of the bad disease outcomes could theoretically be linked to that maladaptive hypertrophy and what comes after that. And so rapamycin as a compound really has a lot of different potential benefits in how it targets that hypertrophy. What we've seen, I think, is pretty promising in the client-owned cat trial, which was either maintenance of the same wall thickness or reduction in wall thickness over time. That was our RapaCat trial. And that trial is now about to be hopefully replicated in a much larger set of cats. So a few hundred cats in what's called the HALT trial, which is about to begin. I think all that being said, there's still some unanswered questions about what we're seeing with rapamycin. So if we dive into kind of the more multi-omic and genetics data that we saw out of rapamycin, the signals at the level of the heart tissue were impacting things like the coagulation cascade, in addition to mitochondrial function and signals that would be autophagy related, all the things we knew rapamycin would do. There's also some other maybe potentially exciting things like coagulation in there in terms of downregulating the coagulation cascade. And so we're now moving on into those. So looking at rapamycin's effect on platelets and platelet function in cats with hypertrophic cardiomyopathy, that's on the docket, and that data looks really exciting. I think that'll be presented here next week or so at an upcoming coagulation conference. But there's I think a lot of exciting work buried in there for us to continue doing to really understand what the possible benefits of rapamycin are beyond just hypertrophy.
Fascinating stuff.
In your rapamycin trial, you use 2 different doses of rapamycin.
Yes.
Because rapamycin may act in 2 different protein complexes. Can you tell us about that? Because that's of course really important. What does the low dose versus high dose might do, and how these different protein complex may affect different pathways?
So in the original RapaCat trial, we used 2 different doses of drug. We called them kind of low and high, but in reality, both of them are pretty low doses. So for the listeners, I think it's important to recognize that high dose rapamycin does very, very different things in terms of immunosuppressive effects. And that's kind of where it was originally used, was in organ transplant rejection, for example. At the lower doses.
It's such a different population of patients, isn't it? It's wild,
Yes.
the different uses. Yeah.
Yes. At lower doses, like we are targeting in our studies, I like to say low dose and ultra low dose. Those drugs are really getting in there and intermittently breaking the mTORC pathways, hopefully writing autophagic flux and altering kind of that drive for hypertrophy that we see in the cell signals at the level of the tissue. And so what we recognize is that as doses go up, off-target effects are off-target for us, that is, beyond just the level of the heart wall thickening, are more common. And so we actually hypothesized that the lower dose would be the dose that we wanted, because we really want to get in there and temporarily break that cycle, let it come back to normal, and then break it again, let it come back to normal, and then break it again. And that's why in our study, you'll see cats get dosed once weekly only at either low or ultra low dose. When we saw the kind of data signals that were coming out of that clinical trial, we then took that data to our multi-omics study. And in the multi-omics study, we actually did a low dose and an ultra low dose that were one step below. And so if you look at our multi-omics study, the best looking data in that study is at the higher of the 2 doses, which is equivalent of the lower dose in the clinical trial.
Okay.
And so all the stars are aligning that there's a sweet spot there for the impact that you want at the level of the tissue, both from a kind of genetic perspective or transcriptomic perspective, as well as in the clinical study perspective. So it is a really interesting drug in that you could do it really wrong if you gave too much of it. And so I am very cautious. I get asked every day if it's time for people to just go out and find some rapamycin and start cats on it. The answer to that for me is a resounding no. I think we need to be sure that if we're using rapamycin and HCM, that we're using a formulation that mimics what was done in the clinical study. So it needs to be this kind of same delayed release compound, and it needs to be at that lowest dose in order to be sure that we're going to be using it safely and hopefully getting the same efficacy that we saw in our study. So I'm right now very optimistic that this is a compound that we'll see used in veterinary cardiology and HCM for years and years to come. I think we're going to uncover new things that it's doing that might be beneficial, and I'm really excited about that. But I'm also cautioning folks that the best way to use it right now is to try and get involved in a clinical study. And that's why we're moving forward with that HALT trial as soon as we can.
Fantastic stuff.
Question you get a lot for sure, I'm sure, Josh, is when to start this drug. Subclinical hypertrophic cardiomyopathy is a very, very wide stage. And of course, maybe potentially up to 70% of cats will never develop clinical signs of their disease, while, I don't know, 1/3, depending on the study, would develop very nasty complications.
Yes.
So when do you think, where is the sweet spot there, the Goldilocks spot? When would we ideally start this drug, at which stage?
Yeah, that is a question that I don't think the data has yet answered for us. I do think the larger trial will help us in stratifying cats, at least between B1 and B2. So in the RapaCat trial, we cast a broad net, and so they could be B1 or B2 in that trial. Similarly, the HALT trial that we're about to embark on will do the same, but the numbers will hopefully be big enough that we'll be able to say, is the benefit the same in those two groups? If it is, could we wait until B2 for all cats so that we're more certain that they're likely to drive towards a clinical outcome than a B1 cat, for example? I think the jury is still out for me on that. Obviously, from a cat perspective, the later we can use the drug while still preventing the bad disease outcomes is the sweet spot, right? So as far as we can push it while still getting all the benefit would be the sweet spot. But it may very well be, given that we're targeting maladaptive hypertrophy, that the best time to use this drug is B1 to prevent cats from ever hitting B2. I just don't know the answer to that question yet. From a physiology perspective, if I were to put a wager on it, I would wager that the drug is going to be most effective in stage B1 and less effective later on in disease. But I actually have no data to back that up.
They're such a hard group of cats to study, aren't they? The B1, just like the B1 mitral valve disease, when the majority of them may never need treatment.
Right.
And a disease that may be very slowly progressive versus much faster progression in a subset. You've got to spot the subset, haven't you? Somehow.
Yes,
and at the moment,
absolutely.
I think it's left atrial size and left atrial function, I think, are our best ways of doing that. But they're far from perfect.
Yes, I 100% agree. And the study that we're about to embark on is really aiming to look at a number of different variables, including left atrial function, to hopefully tell us more about what this drug is doing and where the benefit really lies. But I don't have enough power in my crystal ball to predict it just yet.
It's not yet.
But I'm really looking forward to that data to see where that is. I guess the other thing I think about, though, the kind of cat lover in me thinks, well, if we can show that this drug is really safe at these doses and you only have to give it once a week, and we can ensure that it's affordable, the cat lover in me says, even if only 4 out of 10 cats needed it, I might give it to all 10 cats if I had 10 cats. Yeah. But that's obviously going to be a clinician and client dependent decision, right? Yeah.
And I think at the moment, we treat with clopidogrel in B2, however you want to define B2. We all have our own definitions. And Jose and actually have quite similar cutoffs, but that's by chance rather than consensus. I'm sure we overtreat with clopidogrel. I'm sure we do. And we're all okay with that because of the severity of the outcome, because of how bad an AT is, going back to what we were saying earlier on. We're okay with treating the ones who don't need to be treated, if you like, because we can't spot them. We're not good enough at spotting them to know reliably which ones require the treatment. And I think here, if we have something that is effective, the difference between once daily dosing and once weekly dosing is just so massive, isn't it? To that relationship between the pet and the owner, that surely once weekly dosing's got to be okay for the vast majority of cat owners.
Yeah, I think that's right. I think clopidogrel kind of in and of itself hit that sweet spot, right? The outcome was bad enough. The medication is now affordable enough, and you don't have to give it 3 times a day.
Sure.
And so people have said, gosh, this is worth it in that world of trade-offs. I am secretly very hopeful, though, that the coagulation data coming out with rapamycin will be good enough to say maybe we can avoid doing all of those things at once in cats that don't need it.
How cool is that?
We will see.
What a game changer if that pans out.
Yeah.
And once per week, to me, that's already the owner compliance. We always talk about owner compliance, polypharmacy, blah, 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 once per week.
Yes.
It's just amazing. Yeah. Absolutely amazing. Josh, in your initial trial, you excluded cats with severe outflow tract obstruction. May I ask you, why did you do that? And if you are, and then are you including cats with obstruction now on the HALT trial?
Yes, that's a great question. The real reason we did that is because we wanted to see if this drug actually had a benefit in the general cat population with HCM. We felt really comfortable that any benefit we saw in general HCM would be even better in cats with obstructive HCM. And that's based on the data underlying the reason we ever embarked on this trial to begin with. So we didn't really touch on that, but the data that kind of spurred this work is based on model organisms where obstruction has been surgically induced. And so in mouse models, for example, you band the aorta, you cause pressure overload. And that pressure overload is where rapamycin was shown to reduce wall hypertrophy really nicely. And so if you think about obstruction as a model, as its own form of pressure overload, we assumed that we would just be recreating previously published science by including a bunch of obstructed cats. And so we purposefully avoided that so that we could have a broader indication for the drug, and it might be more useful to the cat population as a whole. I do think we'll pull out some cats with obstruction and do work in them to show that at some point. It is not the purpose of the HALT study, and it is not something that we're currently working on. But in my own personal data bank of studies I'm desperate to complete, I want to try rapamycin in cats with heart failure, and I want to try rapamycin in cats with obstruction. And so I think that there's possible indications and ways that rapamycin could benefit both of those populations. I'd love to do that. I think it would require a smaller study to show possible efficacy there, but haven't gone there yet. The HALT study is really designed to replicate RapaCat and expand on the timeline of monitoring, and expand on the outcomes that were assessed, more echocardiographic features like MAPC and left atrial function included. I do think there's some really cool compounds that are out there in human cardiology that I'm excited to try in cats that I'm hoping we get to. You know, we think about all these drugs that are being used in humans now and showing benefit across every disease known to man. I think hopefully we get some of that done, and I would be willing to bet that those are coming faster than Campton compounds, but
right. And I think the SGLT2 inhibitor family,
that's what I'm talking about.
We've touched on that with Adrian Boswood and talked to him a little bit about it. And last year we had a conversation with David Connolly at ACVIM where we were, all pretty excited because there was the promotion of Boehringer's licensed drug, which obviously is for use in diabetes. But we've got the safety data already for that in cats. Is there potential for a cardiovascular trial? And I'd love to see that on the horizon. It's just another option, another sort of another hope for these cats.
One last question, which I love to ask to cardiologists that are focused on HCM, and in your case as a geneticist also, I would love to listen to have your opinion on. Why do we have such a high prevalence of HCM in cats? 1 in 7 cats. That's outrageous, right? When it affects 1 in 500, 1 in 200 humans. Why the high prevalence in cats? Do you have any theory, any thoughts about it?
Yeah, I think that is the age-old question. I think we don't really know, but I'm guessing that this ongoing genetic work out of the RBC group and out of our group here is going to reveal that there are many, many mutations that are super common and when put together can drive this phenotype. I think the common variant hypothesis is all I'm left with when I think about what could be doing this, because I just don't have any other way to believe that we could look so closely at the genetics of HCM and not find more single Mendelian type mutations. And so I'm stuck in common variant land. I would be thrilled to be proved wrong, but I think until such a time, I'm going with somewhere along the creation of domestic cats as they're being bred and domesticated, or domesticating us as it would be. We're selecting for common variants that are just at the root of being a cat. And some combination of those has led us to the situation that we're in today. And I think in some degrees, we see that in breeds a lot, right? There are lots of very breed-specific diseases that have wildly high prevalences that we've never been able to figure out. And I think the reason for that is because they're baked into the genetics of that breed. It is a fixed variant. And finding fixed variants is something that we are not powered to do.
You can't back up the truck, can you? You can't
Right.
go far enough back to look for where that common variant is or how it came about.
So some really cool evolutionary genetic work would be neat to do. Once we identify what we think these common variants are, taking that to wild cat populations, I think, is something that would be fun, and would be a great project for somebody's PhD thesis that was excited about that type of work. Who knows whether that would be fruitful or not, but it would certainly be interesting.
Absolutely.
You're not going to catch a fish if you don't go fishing, are you? So
Right.
It sounds like an interesting research. Yeah.
Right.
Hey, Josh, thank you very much. It was amazing.
Yeah, thanks for joining us, Josh.
Of course. Thank you, guys.
We like to ask our guests to summarize by giving us the top 3 take-home messages or top 3 key points of the podcast. So if you're happy to share with us what your top 3 take-home messages are, that would be fantastic.
I think my top 3 take-home messages are that there really is a symbiotic or beneficial relationship that veterinary research into hypertrophic cardiomyopathy has with our human pharmacology counterparts. I think cats and HCM are an incredibly great model for helping us understand what's going on in humans and trying out novel therapies. And I think that's going to benefit us in both directions. I think there are some exciting compounds on the horizon, including rapamycin and our myosin inhibiting compounds that I'd love to see come to light in feline medicine. And I think that we have a lot to learn about the genetics of hypertrophic cardiomyopathy. And we're going to get there by creating loads and loads of data and hopefully finding young, inspired scientists to sift through it.
Amazing. Josh, thanks so much for joining us. Honestly, it's been super interesting talking to you. And for anybody out there who's interested in veterinary cardiology, please listen to our other episodes. Please follow us on our YouTube podcast channel and make sure to follow us on socials. It's at heartbeat_pod. Josh, thanks again for joining us. And hopefully we'll look forward to the next stage of your ongoing work into HCM and cats.
Sounds great. Thanks.